Hello, my name is Ashley Reynolds and I'll be doing this project on protein synthesis. Protein synthesis is the process by which the genetic code puts together proteins in the cell. First process of protein synthesis is transcription. It's the first step of gene expression in which a segment of DNA is copied into RNA to produce a complementary strand of RNA. Transcription catalyzed by RNA polymers as well as many steps of this process. First step is RNA polymers recognizes the start site of gene with the help of other proteins. A complex consisting of RNA polymers and other proteins come together and assembles to the DNA strand, begins to unwind a segment of the DNA molecule until the strand begins to separate. As you can tell, that is the strand that's going to be separating in the first step. The second step is RNA polymers uses only one template strand to string together a complementary strand of RNA nucleotides. The base pairing rules follow the same rules as DNA, but uracil is the base, not thymine. As helicase enzymes move a transcription bubble, it would split the double helix into two strands of unzipped DNA nucleotides. By breaking the hydrogen bonds, the growing strand hangs freely until it's transcribed and the strands are zipped back together. Then that one gets transcribed to that one. They're both two DNA strands. The third step is once this gene has been transcribed, the RNA strand breaks away from the original DNA strand. If the cell has a nucleus, the RNA is further processed and exits to the cytoplasm through the nucleopore complex with the addition of 3 UTR polytail and a 5 UTR cap. That is the one that has broken down, and they're both considered the cap, 3' cap, and the 5' cap. 5' cap and the 3' polytail. The definitions that go along with transcription for step number 3 is that transcription produces 3 major types of RNA molecules after all of this is done. The first one is messenger RNA, mRNA, and an immediate message that is translated to form a protein. The second one is ribosomal RNA, rRNA, forms parts of ribosomes. The last one is transfer tRNA, brings immediate amino acids from the cytoplasm to the ribosome to help make the growing pattern. The next step in protein synthesis is translation, which is a bigger step than transcription. It's the second step of gene expression, which during this process mRNA code is used to make protein. Translation translates an mRNA message into a polypeptide. A codon is the 3 nucleotide sequence that codes for the amino acids which will be used in these steps. The first step is the express codon attaches a tRNA molecule that has an amino acid attached to it. The tRNA anticodon pairs with the mRNA codon and brings a new tRNA molecule very close to the other molecule, which is the same tRNA antiparallel to it. As you can tell, them are both antiparallel. They will keep going forward and they will never touch. One's going the opposite direction both go in the opposite direction. The ribosome then helps form a peptide bond between the two amino acids. After that, the bond begins to break because of the ribosome, the tRNA molecule in the second site, and its amino acid. They begin to break apart, which that would be brought into after that would be broke apart and gone. Last, the ribosome pulls the mRNA strand to the length of codon, and the tRNA molecule from the second site would be shifted to the third site, which was called exit site. After that, the tRNA molecule would leave the ribosome and turn to cytoplasm to go through the process again. The other tRNA molecule would now be in the second site, leaving the first site empty, exposing the next mRNA codon, which will be all started over and will go all through the process again. Definitions that go along with this process is stop codons, which is a signal at the end of the amino acid chain. The start codons, which signals the start of translation in the amino acid methionine. Anticodon, the set of nucleotides that is complementary to mRNA codon. Lastly, if you want to join all this together, there are many structures to go along with these processes. Proteins are said to have 4 levels of primary structures: primary, secondary, tertiary, and quaternary. The primary structure is the covalently linked amino acids. The secondary structure is the segments of polypeptides folding the stable structures. The tertiary structure is the complete folding of the protein. Last, the quaternary structure is the association of 2 or more polypeptide chains to form a complex. Protein synthesis, the last definition for this is protein synthesis is a process by which genetic codes put together proteins in a cell. It is like replication in some ways, like how they both store genetic codes. That is all for protein synthesis. Thank you, and bye.